Welcome, and thank you everybody for joining us today for Thieme Chemistry's first webceminar this year. My name is Karen Muirhead and I'm a Senior Executive Editor at Thieme Chemistry, and I work on Science of Synthesis, which is an online reference work and includes a volume on click chemistry, the subject of today's webceminar. Please could you all join me in welcoming Professor Hannes Mikula from the Technical University of Vienna and Professor John Moses from Cold Spring Harbor Laboratory in New York, who are today's speakers, and Professor Floris Rutjes from Radboud University in Nijmegen, who will chair today's event and who'll take over in just a moment. Before we get started though, I'd like to go over a few housekeeping items. Please note that the webceminar will be recorded. Doesn't look like I'm showing my screen. Can everyone see my screen?
Yes.
Okay, sorry. The webceminar is being recorded, and by participating you agree that the organizer may record sound and image and authorize the distribution of the video on different marketing channels without claim to remuneration later. Both the chat box and the list of participants will be hidden in the recording so that no names are recognizable afterwards, and a link to the recording will be sent to everyone who registered shortly afterwards via email, and that link will expire after 4 weeks. Everyone will be on mute during the web seminar, but if at any time you have questions, please submit them to our panel by typing the question into the question window, the query window. And please note that unless you use the anonymous option, your name will be visible to the panelists and may be mentioned. My colleague Alex Russell will be on hand as chat manager and will help in choosing the questions for the panel. And also look out for free content that we'll post in the chat window during the course of the webinar. And also remember to tweet using the hashtag Cheminar and tag us at Thieme Chemistry. There'll be two 30-minute talks today, followed by Q&A for both, with a short Science of Synthesis demonstration in between. And the chair for the session, as mentioned before, is Professor Floris Rutjes, who holds a chair in organic chemistry at Radboud University in Nijmegen in the Netherlands, and his research interests include the synthesis of bioactive heterocycles, click chemistry, and continuous flow synthesis. He's the director of the Institute for Molecules and Materials at Radboud and president of the European Chemical Society, EUCHEMS. Floris, the floor is yours.
Okay, thank you very much, Karen. I guess you can see my screen now and hear me all right. Yeah, let me start then, and welcome you all to this webinar on click chemistry, a webinar that is a nice day, not only because of the date, but also because it sort of marks the publication of a new volume of Science on Synthesis, namely on click chemistry. And now I'm actually very pleased to now see this volume in between all the other Science of Synthesis volumes. So I'm very glad that we reached this point right now. And for that, I first of all would like to thank all the authors very much for their contributions. And they really made that this has become, I think, well, perhaps not comprehensive, but a volume on click chemistry in the style of Science of Synthesis that nicely shows all the developments that click chemistry went through in the past 20 years. And also shows to what it has evolved. Here you can actually see the different topics that we cover in the book. It is more or less also, I think, what chronologically happened over the years. There's a large part on copper catalyzed azide alkyne cycloadditions. That was followed up by metal-free reactions, and later on different variations occurred in the literature, and now yeah, they are more or less logically grouped, I think, in this volume. And then of course in the Science of Synthesis style, where the reactions are accompanied by representative experimental procedures. And in the next 10 minutes or so, I would like to go through some of these topics from a somewhat more personal perspective, as reactions also evolved in our own group, and I also try to put them in a somewhat broader perspective. For me, click chemistry started with a lecture that I attended from Barry Sharpless. I think it was somewhere in the late 90s of the previous century, or the previous millennium, perhaps I should say, where he was raising the question, so if you look at nature, that does many things with one chemical bond and a limited number of building blocks. Look, for example, at proteins. There is one chemical bond, the amide bond, and there are 20 amino acids. And with this simple set, you can create a multitude of chemical functions, catalytic functions, structural functions, etc. The same holds for DNA and RNA. There's only one covalent bond, the phosphodiester bond. There are 4 or 5 bases, and with that you can store all the genetic information. Would there be a way to do similar things, but then with a new reaction, a synthetic reaction, a click reaction, if you wish? And so a reaction that is wide in scope, works well for a variety of compounds or materials, that is easy to perform, insensitive to oxygen or water, and that also goes with a simple workup and product isolation. Now, that was really a conceptual idea, but not so long thereafter, this reaction came up, the copper catalyzed azide alkyne cycloaddition, that has some of those properties. So it is a reaction between an acetylene, an alkyne, and an azide that normally does not proceed so easily. It was published actually for the first time by Professor Rolf Husgen, who unfortunately passed away during this book project not so long ago. And so the reaction would require 100 degrees. It would give a mixture of regioisomers, but if you add a copper-1 catalyst, the reaction becomes regioselective and proceeds at room temperature. Now, this, I think, marked the start of click chemistry, and this reaction was independently discovered by the groups of Morten Meldal in Denmark and Sharpless and Volking at Scripps in the US. And I think one of the reasons, not only for the ease of carrying out these reactions and for the versatility, but there's another aspect that I think is very important that made this reaction very popular, and that is that the triazole that is formed is a very good isostere of the amide bond. I have shown them here. Both, so they are both stable planar molecules. And unlike imidazole, which has the same shape and almost the same atoms, which is relatively basic, the triazole is not basic at all due to this additional nitrogen atom. And one example that I personally like very much that shows the resemblance between the amide bond and the triazoles is an example from the Gadiri group already from some time ago. They were working on those cyclic peptides that, if you have alternating stereochemistry of the different amino acids, can very nicely stack on top of each other and form peptide nanotubes. And if you now replace 2 of those amide bonds by triazole bonds, you see that it is not a real circle anymore. The isostere, the triazoles are slightly more extended, but still, also with these triazoles in there, the molecules nicely stack on top of each other. The triazoles even participate in hydrogen bonding, and you get a functional molecule that has very similar properties to the initial peptide nanotubes. And in our own group, we were also inspired by this reaction. At that time, we were working on glycoprotein synthesis, molecules that are like anchored in the cell membrane with the protein part and have then the sugar part exposed to the exterior of the cell, which is then involved in all kinds of recognition events. And so we have made quite some stable glycoprotein analogs in which we have replaced the natural N-O-acetal or OO-acetal linkage by the triazole moiety. And in fact, at that time, when one of the PhD students graduated, his sister-in-law, Nadine Oudegroen-Badar, she made this little painting, which I got as a present as a supervisor, and which I thought was also a very nice illustration of the book. So it's a sort of artist representation of the stable triazole-linked glycoproteins that we were working on at that time. But then copper, you don't always want to have copper, especially if you are working in living systems. And so then the quest came to design acetylenes that could participate in metal-free cycloaddition reactions. And it was really Professor Bertozzi in the US that moved this field forward, initially with a Staudinger ligation, but at one point she published strain cyclooctines that would spontaneously react with azides. And well, after that first publication, I think quite some groups jumped on this topic and started designing and synthesizing strained acetylenes that could spontaneously react with azides. So some of the examples are over here. And what actually happened is that at the same time, people were trying to look for faster reaction rates, higher reactivity, which is shown here on the y-axis, and at the same time try to keep the lipophilicity of the compounds low, which, as you can see here, did not always work due to the presence of those large aromatic rings in the systems. I think there's this example from the Boons group, or Geert-Jan Boons, I should say on Dutch, which was an early one. There is another one from Caroline Bertozzi that was very fast, and from our group came those two molecules. First, well, we call this one, we call DIBEC, benzoazacyclooctine, but unfortunately nowadays it's known as DBCO, which I think is a less good name, but well, that is the name that is used nowadays, which still has the lipophilic groups. And but soon thereafter we came up with BCN, which was invented by Floris van Delft, an associate professor in my group, and which was easy to synthesize in only a few steps, is not so lipophilic as the ones that have the aromatic groups, and is also symmetrical, which means that you don't get retroisomers into conjugation reactions. So some examples. Here you can actually see the synthesis. So it starts with cyclooctadiene. There is a cyclopropanation reaction, then a separation of the endo and exo isomer, after which the ester is reduced, the triple bond dibrominate, or the double bond dibrominated, and then after a double elimination, the BCN is formed. Now, at that time, we showed that it can be used to color cells. If you have a sugar that contains an azide that is metabolized to the cells by the cells and expressed at the cell surface, so then the azide can react with the BCN if it is equipped with a fluorescent dye. But later on, Floris van Delft, he quit academia and founded the company SynFACTS jointly with my former PhD student Sander van Berkel, and they started actually investigated whether this conjugation molecule could be used in drug antibody conjugates. And now, I think 10 years after the company was founded, there are quite a number of drug antibody conjugates in first phase 1 clinical trials that have this BCN molecule in the linker between the drug and the antibody. And so that also shows that some of those conjugation molecules really have led to clinical applications and hopefully to drugs in the near future. Now, a next step in those bioconjugation reactions are, I think, the cyclooctene, the trans-cyclooctene tetrasin cycloadditions, TCO, that are also being used in linking drugs and antibodies, as you can schematically see over here. These reactions are, on average, a factor of 10,000 fold faster than the azide alkyne cycloadditions. That makes even in vivo click chemistry possible, but also because the functionality is somewhat different, you could also have a click reaction accompanied with release of a drug. Now, I won't say too much about this because I think this topic will be addressed at large by Professor Mikula in the next lecture, and move on to the next slide to present some of the remaining topics that are also summarized in the click chemistry volume. The click reactions that form the other regioisomer selectively, of course, also an important strategy. We have the SYNOpen based cycloadditions that are very versatile and are really a new branch of cycloaddition based click chemistry reactions. We have the thiol-ene and thiol-ine click reactions that are covered, and then also importantly the sulfur fluoride exchange, the Suffix reactions that will be discussed in a lecture of Professor John Moses also later on in this webinar. So these are more or less the topics in this volume. I would like to thank the authors very much again, but last but certainly not least, I would like to thank the editors, the staff of Thieme, which I think brilliantly supported this whole book project. First of all, Toby Reeve. I looked back into the agenda. I visited Thieme at March 25 in 2019, so that is a bit less than 3 years ago. I think the book project took a little bit longer than we anticipated at that time, but then also we had COVID in between. I'm simply very glad that the project finished, and I would like to thank Toby very much for all of his support, and also congratulate him with the little baby that he got today on this beautiful date. So thank you, Toby, and congratulations with the second child. Furthermore, I also would like to thank the rest of the staff and editors of Thieme, and especially for the meticulous editing of all the chapters that were handed in. I think the quality of the chapters was already quite high, but then I think you did a terrific job in putting everything in the Science of Synthesis format, and I think that is also one of the strengths of this book series. And it's throughout the whole series, it's very consistent in the way things are presented. So that is also very nice. Thank you very much for that. And with that, I would like to close this part of the program. And then I have to continue. I stop sharing my screen. Yeah, so my screen is no longer shared, I think. And let me get back to the so I don't see the screen anymore, but I guess you can still hear and see me. So let me then introduce the next speaker, which is Professor Hannes Mikula. After finishing his studies in technical chemistry, Professor Mikula obtained his PhD from the Technical University in Wien, in Vienna, in 2014, where he was already inspired by the field of bioorthogonal chemistry. And that made him join the group of Professor Ralph Weisleder at the Massachusetts General Hospital. and Harvard Medical School as a postdoc, where he spent 2 years. And I think that really also shaped his ideas for his independent career. In 2 years later, he returned to Vienna, where he started as an independent young principal investigator to lead the group on molecular chemistry and chemical biology at the Institute of Applied Synthetic Chemistry. His recent work mainly focuses on the development of bioorthogonal tools and reactions for in vivo chemistry, diagnostic tools, medical imaging, and theranostic applications. Professor Mikula received quite some awards, I would say, in the past 10 years or so. Let me just mention a few recent ones. In the past year, the START Award on the Austrian Science Fund, a Science Award from the Austrian Chemical Society and Austrian Academy of Sciences, a Young Scientist Award from the Federal Ministry of Education, the Karl Schlegel Award from the Austrian Academy of Sciences, and also the Habilitationspreis from the Austrian Chemical Society. And there are actually many more, but I think this will do for now. And I gladly would like to give the floor to Professor Mikula to present on, well, I guess, reactions of tetrasins and cyclooctanes. Thanks. Yeah. So the floor is yours, Hannes.
Many thanks, Floris. I hope you can hear me well. Thanks again for the nice introduction. Certainly no need to mention any award, I should say. So it's all about the science today. No, really, thanks again for the kind words. Also, many thanks to Thieme for this exciting opportunity to, let's say, being able to share some of our recent results of our molecular designs. And that's actually what I'm trying to do now. And I guess that works. Always the two standard questions in webinars and online meetings. Can you hear me and can you see my screen?
Yeah, it's all perfectly fine.
Perfect, thank you. Okay, and as already mentioned, and there will be some overlap with Professor Rutjes' talk before, also some historical ones, I should say. I'd like to talk about tetrasin transcyclooctane chemistry in particular, and the title has been mentioned before. It's all about bioorthogonal bond cleavage reactions, a story that I usually call simply exit the cube, and you will see why this is sort of my favorite phrase to call our research in the past few years. So as mentioned, I've actually been trained as a synthetic chemist, also sugar chemistry, carbohydrate chemistry. So this is something like this is basically what most people have in mind when thinking about synthetic organic chemistry. And our main motivation right now is really to design reactions that work in vivo, so bioorthogonal reactions, bioorthogonal chemistry, or to be actually more accurate, in vivo chemistry. So when comparing these two, right, so in synthetic chemistry, just thinking about all the crazy work out there when it comes to total synthesis, natural product synthesis, it's always really about one specific chemical structure. And the aim is to prepare this no matter how it can be achieved. So we would have all crazy different manifold methods. And this is actually completely different in the field of in vivo chemistry, at least still completely different, where the structure is not that important, let's say. It's actually the how, as we really aim to have reactions that can safely be performed and that safely proceed and selectively proceed in living systems. So it needs to be biocompatible, highly selective, and so on. what's really important to me, and I keep repeating myself in several talks, is reaction kinetics. So those reactions really need to be ultra fast, and I will get back to this in a minute. So why are we actually interested in vivo chemistry? So it's like a standard phrase. So we want to understand biological processes or living systems, but also manipulate living systems. So that basically means it comes to diagnostics and therapeutics. So the design of new approaches, for example, in vivo medical imaging, field that I'm not going to cover today, targeted drug delivery. So that was already shown in Professor Rutjes' slides. So the cleavable antibody drug conjugate will come back soon. Or just briefly calling it, let's say, the on-off control. So can we turn something on? Can we turn something off? And to achieve that, we mainly focus on the design of chemical tools. So similar as Professor Rutjes showed before, I also included a few slides basically showing how I got into this field. And it was a very similar story, although a few years later. So back in 2007, I was still studying technical chemistry, and I just finished, I remember that very well, a course on asymmetric synthesis. And being able to attend this tetrahedron anniversary symposium in Berlin, I still remember I was super excited. I mean, the speaker lineup was very impressive, and it also included Barry Sharpless, which was, let's say, one of my heroes also back then, but mainly because of this course, right? So asymmetric synthesis. So I was mainly, right when he entered the stage and he was about to start his talk, I was expecting crazy structures like this, already sort of getting my brain functioning to imagine 3D molecules. And then, so let's say all of a sudden, he basically showed this. I'd say I was not old enough because I did not get the concept at all. So my first thought was mainly, okay, why triazoles? Why is everybody interested in triazoles? That was the only thing I could focus on. Yes, I understood that this is sort of a very convenient or very efficient reaction, but I completely missed the point that it's actually also all about connecting two things via the triazole. So definitely something I still regret. I could have learned much more from this talk. However, a year later, as I said, I basically started research as a carbohydrate chemist. And back then I was able to attend the International Carbohydrate Symposium in Oslo. And I was like, whoa, that was crazy international meeting, many participants. And it started with an award lecture, and that was the Whistler Award. And back then, the award was given to Carolyn Bertozzi. I was like, whoa, International Carbohydrate Symposium, award lecture. So expecting many sugars. And basically one of her first few slides showed this. And I was like, oh, it's back. Now it can be done copper free. But at this time, fortunately, I was listening carefully enough to really get what this is all about. And it basically totally blew my mind. That was probably the most inspirational talk I've ever attended. And yeah, to cut long things short, it was certainly related to sugar chemistry. So Carolyn Bertozzi showed the, let's say, metabolic engineering of aceto-functionalized sugar precursors into sugar glycan conjugates, so glycans at the outside of the cell, basically proving that the metabolism can, or tolerates the A site, that this is incorporated into glycans, and at different time points treat a developing zebrafish, so growing, let's say, zebrafish with cyclooctanes conjugated to a fluorophore, as I said, at different time points, and then later on really showed that the chemistry is basically happening inside the living fish, and that it's possible at a later point of time to really take a closer look on those glycan structures, also in light of the development process of the fish. So that's certainly one of the landmark papers, if not the landmark paper, in the field of bioorthogonal chemistry. And as I said, completely changed my view on this topic. And that was basically when my fascination started. So we've already seen quite some cyclooctines before. I'm bringing several of those back here. So starting from the original designs, there was a lot of work invested, I can only imagine. And I'm really sorry for using the wrong abbreviation here. So it's DBCO in BCN. But the good thing about these two molecules, I'd say, developed by Professor Rutjes and his team, is I would say that these two are still the most commonly used and the most efficient ones, also commercially available, I should mention. Yes, there are some faster cyclooctanes, more reactive ones, but in general, we could basically say that the second order rate constant is in the range of 1. Some other examples now that boosted this kind of chemistry up to 10 or even a little higher. But for, let's say, these cyclooctanes shown here with the A side, it's in the range of 1 per molar per second. So it's always like this, okay, what does that number mean, and why do reaction kinetics really matter? So just to discuss that on an example, so let's imagine we have this reaction of A plus B, and that gives C, so a classic click reaction. And we would consider different concentrations of A and B, and now want to know, okay, how long does it take to achieve 95% conversion, just as an example. And we would go with the rate of 1, then you can clearly see that in a micromolar or even sub-micromolar range, which is basically the range that we need to work with under in vivo conditions to keep the chemistry safe, then it's obvious that this would take certainly forever, especially when the application is, let's say, a time critical one. And as we move up here with the second order rate constant, of course, this becomes more and more feasible. In addition, what I also like to mention is that, of course, a living system is simply not a flask. So, for example, you could argue, yeah, I don't really care whether it takes 19 seconds or 30 minutes. Especially as a synthetic chemist, right? You would usually say, yeah, it's nice if it's done in 5 minutes, but if it's done in 30 minutes, it gives me enough time for a coffee. Or I simply go home and leave it overnight or whatever. However, that's simply because that's sort of like a confined system, a flask, constant reaction conditions. And this is certainly not the case for a living system. So if a reaction is not fast enough, that basically also means that, for example, if we have one molecule that is already, let's say, pre-targeted to a certain position in the body, there is a second molecule sort of like coming by, and we have local concentrations, of course, and for the reaction to occur, this basically needs to be fast. Otherwise, it's not like, okay, let's wait half an hour longer and maybe it clicks. No, it might simply pass the other reactant and not react at all. It might get excreted, degraded, metabolized, and so on. So basically, not fast enough would actually mean it's not happening at all. So pharmacokinetics, right, to come up with a more general term, definitely also has an impact on reaction time. And also mentioned, right, the game changer in our field certainly was the tetrasin ligation or the tetrasin transcyclooctene ligation, as we were certainly in desperate need for speed. So that's just briefly, so-called inverse electron demand Diels-Alder reaction. Inverse simply because it was the second one probably discovered. So it's the inverse electron demand Diels-Alder reaction. It forms these dihydropyridosine click product. You would usually see it like this. Maybe a different tautomer is drawn, and in figures it often says, yeah, plus isomers or only one isomer shown, as usually no one really cares what's going on in this system as long as A and B are connected. I mean, that's the purpose of the click reaction here. The IETA reaction basically is actually only the first step here. So the Diels-Alder cycloaddition, that's followed by a retro Diels-Alder. So eliminating nitrogen, finally forming this system. So of course, it was known from the very first paper. So Professor Joseph Fox, who is definitely the inventor, let's say, of this chemistry. And it was known from the beginning on that this system here is prone to tautomerization. So we can have two different tautomers that are sort of in equilibrium also with this, yes, let's say initial click product shown here. And over time with oxygen, especially under physiological conditions, this can then oxidize to an aromatic system. Most importantly, yes, it's this 5, I would say up to 6 orders of magnitude. This reaction is substantially, significantly faster, I'd say, than any other bioorthogonal reaction. So with second order rate constants of 100,000 and even higher. So you can go higher with more strained transcyclooctanes. I'm not going to show these. This is when it becomes tricky with in vivo chemistry because stability, of course, also plays a huge role. So 100,000 is definitely a good number here. And I should have inserted a reference. So we have recently actually shown with our colleagues in Copenhagen that a number of around 50,000 is a very good, let's say, indicator for successful in vivo ligation, even in short time frames. So the reaction, of course, is biocompatible, highly selective, non-toxic, and so on. So this is what really enabled in vivo chemistry. In addition to the transcyclooctane, of course, the reactivity also depends on the tetrasin. I'm briefly going to talk about this as we need this later. The reactivity of tetrasins usually, or in general, increases depending on the substituent from alkyl to aryl. Electron withdrawing or electron poor heteroaryls that I would say have similar reactivity than, as we say, H tetrasins, so monosubstituted tetrasins here. So of course that means there are plenty of different combinations possible, and I'd say the aryl H here and the bis-pyridal tetrasin, these two are definitely the most widely used ones when it comes to very fast tetrasin ligations and especially in vivo chemistry. The nice thing about tetrasins, despite their cumbersome synthesis, is those compounds are usually bright pink, maybe to red colored compounds, which makes working with these substances sometimes really colorful. And that's also an important side note, as I'll get back to this later. Also mentioned already, and that's sort of the main motivation of my talk, is the advancement of tetrasin TCO chemistry to bond cleavage reactions. So as shown already before, so we can react a tetrasin with a transcyclooctane, as we later can call it the release TCO, which is modified here in the allylic position. This will undergo a classical tetrasin TCO ligation, and then depending on the two different routes for tautomerization, as shown before, form different isomers, where one of these two, and that was hypothesized from the very beginning on when Marc Robillard, the inventor of this chemistry, published about this reaction the first time. So that was in 2013. So one of these two can undergo a 1,4 elimination, finally leading to release of this carbamate moiety. Following elimination of carbon dioxide releases this primary amine, as shown here. And that reaction from the beginning on was termed click to release. There are several others now called click to release, and this is similar with click chemistry, sometimes hard to come up with a very valid term in definition. However, the main motivation for this chemistry from the beginning on, is also already shown in Professor Rutjes' slides, and envisioned by Mark Robillard from the beginning on, was to use this chemistry for the design of ligand drug conjugates. So for example, antibody drug conjugates that, as you know, are basically used to shuttle toxic drugs to the site of disease. So for example, a tumor. And the activation of these would usually depend on, let's say, tumor biology, uptake into cells, enzymes, pH responsive stuff, and so on. And Mark's, so who's basically, or actually running his own company, TechWorks Pharmaceuticals, motivation was to introduce a cleavable TCO in between that antibody and the drug, so that at a later point of time, it is possible to use tetrasins, let's say as bioorthogonal scissors, to cleave the drug off. So which would make this system no longer dependent on tumor biology. So it's also possible to use antibodies that, for example, target receptors that do not internalize into cells. And of course, what's very exciting for everybody in the field is that a different approach, let's say, but certainly the same chemistry is used by Chasqui, so a US-based company, since at the end of 2020 or early in 2021, finally in clinical trials. So this chemistry is, let's say, finally happening in humans, which basically marks the beginning of the era of, yeah, let's say, clinical translation of bioorthogonal chemistry in general. So the problem with click-to-release chemistry is there is sort of like even more need for speed and efficiency, right? In the field of click chemistry, we are used to say, okay, we have A and B and it's complete conversion and everything is fine. So in this case, it's not only the click kinetics, so these two compounds need to react with each other, so otherwise nothing happens at all. But following the initial click reaction, of course, the release kinetics play a crucial role. And as you will see, the overall yield of this process, is, at least in the beginning, was far away from being 100%. So the cleavage yield or the release efficiency is an important parameter, and of course also bioorthogonality. And the key problem at the beginning was that when we look at different tetrasins and we go up with the reactivity to, let's say, achieve sufficient click rates here in step 1, that we drastically go down with the release yield. So when using commonly applied Pyridal Tetrasins, the release at the beginning was, I'd say, only poor, definitely lower than 10%. And that brings me to this graph that I will use also throughout my talk, where we, let's say, put the release yield and the reaction rate of the initial ligation step, and would say, yep, actually where we want to be is up here. So definitely, ideally in the range of 10,000 or even higher in the release yield of 90% plus. And when I started working on this, and that goes back to my time as a postdoc, we were actually wondering at the beginning, okay, so why is release not complete, right? Why is it incomplete at all? And at that time, it was like the dimethyl tetrasin, DMT, was sort of the tetrasin of choice to achieve, let's say, around 80% release. And when looking at this reaction, we were wondering, okay, if this forms the initial click product, and there is an equilibrium between the two tautomers that we call B1 and B2. After, let's say, quite some time, there should be release only. I should also add here the final tautomerization here, which leads to the aromatic system, is no driving force of the reaction. And hypothesized from the beginning by Marc Robillard was that there is also oxidation going on of this non-releasing tautomer, and that sort of like leads to release yields lower than 100%. So we did this reaction, even let's say no oxygen at all, no oxidants or any other stuff, and we were never able to achieve 100%. So we started with quite a number of evaluations, and I'm not going to go through all of these. What we basically learned back in those days is that this reaction basically depends on everything. So which buffer is used, how concentrated the buffer is, and most importantly, also pH. So at physiological pH, as you can see here, we basically found out that the release is sometimes limited to only 20%. And even, right, when we started this reaction at the higher pH, for example, pH 9 or 8, and let it steer, let's say, for a while, and then went back to acidic conditions, we could not improve release. So we were like, okay, something is going on. HPLC or LCMS clearly showed us it is not an oxidation happening, as the mass was always related to B2. So we were wondering, okay, why is B2 forming, and why is it sort of like suddenly becoming a stable tautomer? And the reason is actually that it was not B2, as we were finally able to show. And yeah, let's say unravel the reason for incomplete release. It's always hard when drawing something in, let's say, in 2D, as we're all used to do this, because what's actually happening here is that this initial click product and this carbamate functionality, they can undergo a nucleophilic addition and form, as we call it, a tricyclic dead end tautomer. And this one is no longer releasing. Sounds a little weird, or looks a little weird to draw it like this, but when looking at this, let's say in 3D, using model compounds, you can clearly see that this is actually, let's say, not a far distance to achieve this. So knowing of this tricyclic dead end, we basically added it to this reaction scheme as shown here. And when doing this, that was sort of the first time when we noticed, oh yeah, that basically looks like a cube. And this is when we came up with this, let's say schematic saying like, this is the release cube. And we like working with this because the top layer were sort of like the intermediates, where the bottom layer are the final products. We would have the front face here, which let's say is the bad face, the no release face. The face in the back is the release phase. And I will get back to this later because some of you might already notice that, let's say, the release cube has a flaw. However, that is, basically when our journey started to, let's say, exit the cube. So I'm not sure whether you are a fan of exit the room games, or during the pandemic, I'd say many board games became popular. I need to admit I'm totally into this, and that's why it was basically 1 + 1 = 2 that we came up with this phrase. Okay, can we exit the cube? So how can we circumvent any problems and really boost biothermal bond cleavage chemistry? The first very straightforward idea was, can we avoid the formation of the dead end, which was as I said, straightforward, because when blocking this nucleophilic addition here with a methyl group, that was the idea back then, and it was not surprising, knowing now what's going on. It was not surprising to see, yes, we can definitely achieve complete release, so complete, let's say 99% plus. However, that was with alkyl, so bis alkyl tetrasins only. So what we basically achieved is we moved this pink square here, let's say up from, depends on the conditions, from 20 or 80 up to 100, meaning we're still much too slow. So basically next step was like, okay, yeah, can we accelerate this reaction somehow? So that basically brings me back to this initial tautomerization process. And we were like, okay, how can we trigger this? And how can we accelerate also the release step? And that goes back to our very first report when we came up with tetrasin acids, actually originally motivated by the fact that release was faster under acidic conditions. And later, Sander van Kasteren came up with a scaffold that I need to admit is way more powerful. It's these ammonium functionalized tetrasins. And what these, let's say, directing groups basically do is, after or upon the click reaction, these groups direct tautomerization, so to accelerate formation of, as shown here, the releasing tautomer, and moreover, it also enhances the elimination step. However, and this is something that always needs to be considered, we would basically have two different click orientations of the tetrasin. So if the acid, as it's shown here, is, let's say, close to the leaving group, it would direct formation of the releasing tautomer and really lead to fast release. Whereas if it clicks the other way around, it would basically favor formation of B2. There might be some equilibrium going on, but the release is definitely only slow. So, and considering this, we have basically termed these two click orientations head-to-head and head-to-tail. Need to admit that was basically the best we could come up with. So when speaking of accelerating, we were like, okay, can we use aromatic tetrasins? So heterocyclic tetrasins. And just quickly switch here. So hope you can still hear me well. It seems to work. So we were basically wondering what's going on with aromatic TZ and why are these not properly working in release experiments. We made these alkyl heteroaryl tetrasins and investigated release. And what we could basically see was, a release of around 50%. Yes, with the acid, as it's shown here, it's basically instantaneous release, right? This is what the acid is doing, but then it is still stuck at 50%. And that's basically what I've said before. So it depends on the click orientation. So if the alkyl here is in head-to-head position, then we achieve release. Depending on whether it's a methyl or an acid, it will be fast or medium. And as soon as the aromatic system here is in head-to-head position, then we sort of get stuck at B2. And we could show that this B2 is no longer in equilibrium with the initial click product. So it is really a stable compound that we can isolate and analyze by NMR. So it's this 50-50 around ratio of click orientation that is a key limitation, as we simply cannot control the click orientation. So we thought, okay, can we outmaneuver click orientation, and came up with the C2 symmetric, yes, it really is C2 symmetric TCO that has these two leaving groups here in a allylic position. And with C2 TCO, as we call it, if we would react this with the TZ that has one directing group, it would no longer matter whether it clicks in this direction or in the other orientation, the directing group will always be in head-to-head position to at least one leaving group, which is why we thought, okay, then we should achieve rapid tautomerization, elimination, and basically instantaneous cleavage, say omnidirectional cleavage. And I'll get back to this, of course, as we cannot control which of these two molecules is cleaved. So we thought, okay, this could be fast and complete, independent from the click orientation. And long story short, this is exactly what it is. So we could show this for several tetrasins that we achieve basically quantitative release in short time. So we'll see an example soon. And I also want to highlight that we basically also included a Pyridal Tetrasin. So that was, as you can see, a few years ago. And the release here was still poor, which we thought, okay, that would maybe suggest that the tautomerization is not going on at all. So there can't be any wrong tautomer formed, as we would have two different leaving groups. So we took a closer look and basically found out that it is actually tautomerizing. It even forms to some degree, let's say, the right tautomer, but the right tautomer is still not releasing, or we would only achieve poor elimination. And that was sort of an important information back then, and I will get back to this later. The initial problem for C2 TCO actually was that by introduction of a second allelic modification, we created more steric hindrance, which basically slowed down again the initial click process. So we thought, okay, we can also accelerate tetrasins by basically putting no substituent there at all. So an H tetrasins that were before that or still known as the worst when it comes to release. But in this case, we thought, okay, this should not really matter as long as this R group here is a directing group. So we would still achieve cleavage here. And we used Sander van Kastelen's, let's say, H ammonium tetrasin, here shown as the amine. And yes, that was something that worked really nicely. So again, achieved complete release now with a rate of almost 400, or second order rate constant of almost 400 per molar per second. But just to put this in perspective, that means 10 micromolar TCO plus 50 micromolar TC would mean 99% cleavage in 5 minutes. And that's the rate determining step here, is the click reaction. So we cannot sort of delineate the release here, as it's basically instantaneous. But of course, the problem is we have no control with C2 TCO of which of these two balls here is released. So, and that was also not the intention of this study, because what we aimed for are turn-off applications, right? I've mentioned at the beginning, on-off control. And what we did, for example, to turn off, so we put the C2 TCO.
Hannes, I'm sorry to interrupt, but could you are you coming to a close soon?
Yes.
Yeah. Okay. Thank you.
Sorry for that. So we put that in between. We labeled cells with this, and later edit the tetrasin and could show that we basically cleave off the fluorophore here completely, and it gets dark again. So why would someone do this? Then you might want to look up the concept of multiplex cellular imaging, and there is a very nice study hopefully coming out soon that we're actually very glad to have participated. However, so to finish this up, basically we were still exiting the cube. So we thought, okay, if the TC acid is improving release and the pyridal TC is improving click, so why not combine this and create a phenolic one? So let's say hydroxypyridal tetrasins, and our hypothesis was that this acidic group also controls tautomerization and later on helps with the elimination. So Martin, back then one of my PhD students, now postdoc, made this molecule. Still remember, he put it into aqueous solution and it became orange, yellow-orange, which basically we thought, oh no, it simply died. So we added a TCO and yeah, realized, oh, wait a second, it decolorized. So maybe there is some chemistry going on. And in fact, it later turned out that it is, and that even the elimination works very well. So we achieved basically complete release in 90 minutes. This is how long the release took, which is basically due to a competition of these two OH groups, which means we cannot really control that only the right tautomerization is going. So we were very pleased with this, as this is sort of the first bis-pyridal TC that can really be used for bioorthogonal bond cleavage. And we could move up the click rate to 1,300, which brought us quite close to this green shape here. And to finish, there's one last thing that I want to mention. So thinking of this tautomerization process, we basically then thought, okay, can we control this via the TCO structure? And inspired by Milan Fraber's group, we thought, okay, can we add this OH group here at the TCO scaffold that will later control the tautomerization, form the right tautomer, and eventually lead to release. However, in the meantime, we realized that even with pyridol, even if we get the right tautomer, there is still no release happening. Which means, okay, if we achieve this, we might still end up with a non-releasing system. So, however, Walter, PhD group also, PhD student also in my group, he was already, let's say, too far to quit. So he made this molecule, and we thought, okay, then let's test it. So he clicked it with the bis-pyridal tetrasin, and we thought, okay, yep, we should get the right tautomer, and maybe we end up with a compound so that we can study what's going on here. And he came back to my office and said, no, it's actually done. So we have complete release. And I was like, okay, how fast? And he said, no, the first sample. We're like, oh no, that's impossible, because we know that, right, if this clicks and we get the right tautomer, the 1,4 elimination cannot happen. And in fact, this is what he showed. There is no 1,4 elimination, as what's going on here is basically a new mechanism. It's as we call it, an intramolecular cyclization. And due to this intramolecular control of tautomerization and elimination, we called this molecule the ITCO, which with bis-pyridal, just to put this into perspective, has a second order rate constant of 10,000. The release is instantaneous, it's complete. And as we know now, it's also very, very stable. So just an example, 3 micromolar plus 6 means it's complete in less than 5 minutes. And we finally ended up here in this green area, which we are actually quite happy about. And to wrap this up, remember the cube, I said there's a flaw that bothered me from the beginning. Yes, there's of course a corner hidden here in the back. And I'm happy basically to say that this is sort of our new release product here, which is formed from this tautomer here. It is part of the release phase. So we finally, let's say, found the exit from the cube, which is basically the back door that has been hidden so far. And with that, I like to thank all the people who really do the work here, all my team members, of course, also the entire group, my colleagues, and also the young students joining us. Special thanks also to Jonathan Carlson, a genius when it comes to chemistry, biology, and medicine. Very glad to have him as a friend and collaborator. Of course, also thanks for the funding, and finally to you for your kind attention, and sorry for going over time.
Okay, thank you very much, Hannes. I see that the molecules that are behind you, you even have bigger ones of them, like you showed here on the pictures.
Yeah, but those are made out of plastic. I need to admit.
Okay. Well, thank you very much for sharing this, I would say, intricate details of release in those cyclization reactions. Meanwhile, some questions have come up. And so one question that I see is, are your bis heteroaryl tetrasins, are they planar molecules?
That's a very good question, because that's sort of part of the hypothesis also when it comes to deprotonation, why those molecules are no longer pink. In fact, we cannot say whether these compounds are still planar, but computational investigations would suggest no. But there will hopefully also another paper soon come out that will show that a bis Pyridal Tetrasin, so no OH, is not planar either. So yeah, basically hard to answer the question.
And another question is from a person who considers himself a complete outsider in the field. Would it be easier to make the diacid of the tetrasin instead of making the allyl derivative?
Yeah, I skipped that part and still went over time. So we made the diacid already, that was already included in the first paper. That achieves complete release. It's similar to the, let's say, yellow tetrasin, but the click rate is really low. So it was the first TZ that achieved complete release, but the click rate was, I guess, yeah, less than 10 per molar per second.
Yeah, I guess that answers the question. Perhaps one last question from my side. So of course, there are advantages to being able to release compounds, being able to release drugs at will in a living system. On the other hand, it requires two APIs that need to be approved by FDA or EMA or whatever. So how do you look at that?
That's our enemy question always, and I can only cite or quote basically Mark Robillard and also from their papers. So if you would consider the ADC approach, and you would have, for example, 10 different ADCs, all these ADCs could be, yeah, if you're lucky, cleaved with the same scissors at the end. Which means for 10 drugs, it wouldn't mean 20 compounds, it's 11.
That would, that would certainly help. And on the other hand, or also to support that this is a viable approach. I mean, like you mentioned, there is currently an ADC in clinical trials, which also shows that it must be a feasible approach indeed. Yeah.
And I should also say, right, just it's always costs and tox study and translation. But at first, from a scientific point of view, I think what's also important is the question on whether we can do this, right? If you would go back 40 years in time and compare to what we have right now, then you would need to cancel many things because it didn't seem feasible back then.
Yeah. And then maybe a last one that comes up that says, you made no comment on the difficulty of the synthesis of tetrazines. Could you use different heterocycles used for click chemistry to apply the release you want?
Yes, I made no comment on how tricky it is to prepare tetrasins, or how tricky it is to prepare transcyclooctanes. You know that even better than I do. But yes, definitely no time for that. Other heterocycles, there are triazines out there, but as far as I can tell and as far as I know, there is no other heterocycle that would achieve similar rates than tetrasins. And yes, the synthesis is not easy, but most of them, at least most of them, are pink. So you saw the column, it's usually, yeah, I'd say easy. People here would kill me, sorry, to get those.
Yeah. Okay. Thank you very much. Well, synthetic difficult it could also be an opportunity, because that still requires that we come up with better methods.
There are
those compounds.
Several new methods out there, mainly from Joe Fox, that are really great, that made our life much easier.
Okay, thank you very much for presenting, Hannes.
Thank you.
Then we will move on to a brief intermezzo, which will be presented by Karen Muirhead of Thieme, who will give a short demo on the eSOS system content. So Karen, the floor is yours.
Thank you, Floris, and thank you, Hannes, for the interesting talk. Science of Synthesis is the largest collection of organic synthetic methods available worldwide, and it gives you access to more than 20,000 reliable synthetic methods. It's written by chemists who are experts in the area that they're writing about. And so the reactions and the structures that they've selected give you the best reactions. As it's a full text resource, the advantages and drawbacks of different approaches have been discussed, and the authors have added their unique insights into the science. Science of Synthesis is unique in that way that it provides critical reviews on synthetic methodology and organic and organometallic chemistry. So ultimately, this can save you time when you're planning a synthesis by giving you ideas for how to tackle a difficult step in your synthesis, or maybe you're just looking for a good and reliable method for a particular transformation. If I scroll down here, you can see that the core content is organized systematically and logically based on the product, so specifically the main functional group that you want to make, and it's structure and text searchable online, so text searchable here and structure searchable there. As it's a series of reviews, it can be particularly useful also in getting background information when you're starting a new project or a new stage in your project, and it's also useful for teaching and for consulting. To complement the core product-based structure, the trends and innovation here in the reference library show they focus on the methodology and important techniques and technologies, and this is where we find the Click Chemistry volume. Just click in there. You'll see here introductory information on the more traditional click chemistry and its applications, and just here, see what sort of things that are in there. The reference library topics are also mainly focused on synthesis, but not exclusively. They give a lot of background and introductory information on principles and practices. For example, if I just go into the tetrazine section from Professor Mikula here, there's lots of information on the reactivity and stability of the tetrazines, like he showed before in his talk. It gives Science of Synthesis presents chemistry in context, so each full text article has the same basic layout, and that makes SOS user-friendly. So as well as discussion of synthetic methods, the scope and limitation of reactions is discussed, and where appropriate, there's also preparative details for the reactions. So I'll just show that in from Professor Moses here. Got the discussion and then appropriate experimental procedures. Links actually to both Professor Mikula and Professor Moses's chapters have been put in the chat, and these chapters are available for free until March the 8th. And I'll now hand back over to Floris and to Professor Moses for his talk.
Okay, thank you very much, Karen. Yeah, I must say we also have the e version in our library, and actually I also actually use it in some of the courses that I teach at the master level, which gives the students very easy access to all kinds of details of actually a large variety of reactions. So it's also very useful for teaching, I have to add. Okay, well then let me introduce the next speaker, Professor John Moses, who originally is from Wales, or perhaps I should say North Wales. Studied chemistry in Bath, and then did his PhD in the group of Professor Jack Baldwin at the University of Oxford, and in 2004 moved to Scripps, where he joined the group of Professor Sharpless on the development of click chemistry. And so the name of Sharpless seems to be a sort of common theme in our careers at one point. Recently, Professor Moses joined Cold Spring Harbor Laboratory as a professor of chemistry, and well, there he continued his work on click chemistry. So new findings are something that is called accelerated Suffix click chemistry, and also a new drug discovery method termed diversity-oriented clicking. I guess we will hear about this in a minute. Professor Moses has also spent significant time in helping to build collaborations in the developing world, which I think is noteworthy mentioning. In 2011, he was appointed as a distinguished visiting professor at King Saud University in Saudi Arabia, where he mentored scientists at the Female Chemistry College. And in 2012, he established the Amity Institute for Click Chemistry Research and Studies at Amity University in India. And currently he is the honorary chairman there. And so a key goal of him and his institute is to recruit female scientists to the field of click chemistry and provide a pathway for students to engage with research. So that is really an important societal activity, I would say. And then more recently, you also may have heard from Professor Moses, as he is one of the recipients of the 2021 Organic Division Horizon Prize of the Royal Society of Chemistry, which is called the Robinson Award in Synthetic Organic Chemistry. And this is really a team science award that was given for a project on multidimensional click chemistry, which we also may hear more about. I would say jointly with his previous supervisor, Professor Sharpless, and also Professor Hans Zuilhof from the Netherlands, with many more people that made part of this team. Well, that was already quite some introduction. I won't take any more time of you, John, so feel free to take the screen and move ahead.
Thank you, Floris, for a great, kind introduction. I'll just share my screen. There we go. Well, first, I'd like to thank the Thieme team for organising this event, and also to Floris for editing this wonderful book on click chemistry. It's a real joy to be a part of that, and I thank you for the invitation. So today's talk is going to be about diversity-oriented clicking and how we use that to discover functional molecules. Now, really, this is, I guess, an ensemble of different click reactions. I think really a number of things will be covered that have already been introduced by Floris and Professor Mikula earlier on. So just a brief overview. I'm going to introduce a new concept we developed called the plug-in concept, and then move on to diversity-oriented clicking, and just mention some cycloaddition chemistries and some Michael type style chemistry, building out from a core hub. And you'll understand what that means once I get to those slides. And then just introduce some functional molecules that we've discovered using this technique. So I'll go back to front and acknowledge my collaborators, just in case I forget to do it throughout the talk. But first of all, I'd like to thank my team in Australia, my postdocs and PhD students that worked very hard to help establish this chemistry. Some of my new colleagues at Cool Spring Harbor, Josh and Rebecca, who are also joined the click chemistry team recently, and my collaborators at Scripps, Barry and Dennis Warland, really, who contributed to some of the work that I'm going to talk about. I'm going to dedicate the lecture to Professor Huistjen, as Professor Rutjes mentioned, passed away recently during the preparation of this book, who really was a pioneer in cycloaddition chemistry and really click chemistry, I guess, albeit perhaps not in name. And this wonderful photo, I had the opportunity to meet with two of the greats at Munich Beer Festival, the Oktoberfest. So I took this snap, and I'm very glad I did. Unfortunately, it wasn't the best photo, but it's a historical photo, I think. So what is click chemistry? Well, I think it's a really democratization of synthesis. And if you go back to the original manifesto, which is the 2001 click paper that was written by Sharpless, Finn, and Kolb, really was this click chemistry was inspired by this this fact, or this publication by Gieder and co-workers, that estimated the number of possible drug-like molecules in in the universe, or in the potential universe, limiting to these kind of atoms here and below 500 Daltons. And it turns out, this is just an astonishing number, about 10 to the 63, and that's probably an underestimate. So with that number, Barry just kind of put his head in his hands, and I thought, my goodness, there's just so much to discover. What are we doing? We should just really restrict all searches to molecules that are easy to make. And this is really a guiding principle of click chemistry, I think, an under-recognized principle. And in Barry's own words, chemistry, it should just be simple. Keep it simple, sweetheart, you often hear him say. So I think that really is click chemistry in a nutshell to me. What it's become in other areas, bioconjugation and material science, has been incredible. But I think this really goes back to grassroots, where click chemistry, the concept was conceived. So what is it? Well, it's been introduced already. Click chemistry is just a philosophy, some guiding principles and ideal reactions that should be used in order to make functional molecules. And these kind of rules or criteria for click reactions, high yielding, modular, widened scope, so on and so forth. Really, this is like molecular Lego. So we want to take modules and clunk them together reliably. Once you hear the click, it's done. That's how it should be. That's the basics of click chemistry. Already introduced is the QAC reaction, discovered independently by Meldal and Foken and Sharpless in 2002. And that's really been the cream of the crop. And I think that's taken over from the original fundamentals of click chemistry, because the original paper manifesto, this reaction hadn't been discovered then. So the basics of click chemistry were established before the QAC reaction came along. But this really overtook everything, and I think it's overshadowed everything until perhaps the Suffix reaction, which was I guess discovered or developed in 2014. This is another manifestation of click chemistry. Rather than a cycloaddition process, it's just an exchange. So you're swapping a fluoride for an oxygen in this case, and perhaps generating a stable silicon fluoride bond. This is a really powerful reaction too. So I think while there are other click reactions, I think these are two of the perhaps dominant ones in synthetic chemistry at least. So why sulfonyl fluorides? I'm going to focus more on this style of click chemistry. Sulfonyl fluorides are quite special. They're unusually stable, I should say, but they can be activated through catalysis. Now that's the essence of Suffix chemistry. And if you just look at some of the properties, I mean, they're very stable to nucleophiles generally, homolysis, they're stable to homolysis, high temperatures. They're just unusually stable compounds. And here's an example of a reaction sequence that we did on another project where we took this aromatic nitro sulfonyl fluoride compound. We reduced the nitro group, did a diazotization, then a classic click reaction. That sulfonyl fluoride group was untouched. I mean, if that was a sulfonyl chloride, I think we'll all agree that wouldn't perhaps survive those conditions. But it just kind of shows you how stable these compounds are, or these functional groups are, I should say, until activated. And in this case, we took trimethylsilyl azide with a bit of DBU, one equivalent in this case, and we managed to get the sulfonyl azide. So that you can activate them. So they're almost sleeping beauties, we call them. So the plug-in concept, this is going to be, I guess, fundamental to my talk. Well, it's very simple. We liken it to these kind of electrical towers where you just plug in different units or modules, and whatever modules you put in will determine the function that you get out. And it's all about discovering functional molecules or obtaining function. We wrote about this in a ChemSot review about 3 or 4 years ago, excuse me, and it covers this concept quite nicely in detail, and also in the book chapter that we discussed earlier. So the connectors, I mean, this is nothing new. Nature loves connectors, and of course, DNA is the greatest, phosphate here. This connector is one of the great connectors of all time. We wouldn't exist without it. But also peptides, amino acids can be seen as connectors. You have a nitrogen on one end and a carboxylic acid on the other, and it allows you to connect molecules, well, indefinitely. Same with these carbohydrates, the connections from one oxygen, or one side of the ring to the other, etc. So the connections is what nature really exploits. And of course, click chemistry is inspired by Mother Nature. But what do we mean about suffix connectors? So, well, the concept is that we can take a sulfur center with fluoride substitution, generally high oxidation state, and hopefully we could substitute each of those fluorides. Now, this SF6 would be the greatest connector of all time, I think, but unfortunately it's just too stable. So we have to settle for more reactive sulfur fluoride compounds. This thionyl tetrafluoride is the multi-dimensional connector that Floris mentioned earlier on. I'm not going to go into too much detail about this, but another connector here is sulfuryl fluoride. Now, this is available on ton scale. It's like $1 a kilogram, I think. I mean, it's so cheap. but it's a great connector because we can substitute each sulfur fluoride bond sequentially. Now, these are the inorganic connectors, well, I guess we call them, but there are also other suffix connectors, more the organic ones that we kind of call them locally at least. In this case, they're appended to an unsaturated or double bond. In this case, this is ESF, and this is bromo ESF. So you can imagine doing sort of chemistry at this region of the molecule, and then doing orthogonal chemistry at the sulfonyl fluoride end. So I'm going to focus on this kind of style of connector today. But first, I'm just going to show you an example of the power of connectivity, why we're really focusing on this. So here's SOF4. You can react that with a primary amine to get these amido oxidized fluoride, sulfur difluorides, and these are very bench stable, I've got to say. This gas is very reactive. You've got to be careful with that. But once you make these difluoride amido compounds, they're very stable, bench stable. Well, of course, they've got a sulfur fluoride bond, and we know that suffix, its chemistry is possible with aryl silyl ethers. So if we mix them together with some DVU catalyst, then of course it's set up for polymerization. Indeed, that's what you get. I mean, this reaction goes 99% yield. This is an incredibly powerful reaction. But of course, we're left with one sulfur fluoride bond at each end of the molecule here, or each end of the monomers, you should say. Of course, what can we do with that? Well, it turns out that if we just switch the catalyst to a stronger base, then we can actually do a suffix reaction on the polymer backbone. Now, that's quite difficult to do, but in this case, it works very well. We get 95% recovered yields generally. Well, we don't stop there. Once you put in one module, then we have, we've got an alkyne handle here. Then of course we can do classic QAC here. So just another step, we take AZT, this classical HIV anti-HIV drug, and we've clicked it onto the surface of a polymer. So in 1, 2, 3, 4 steps, we've got a completely new material, an unprecedented material. But more than that, we actually have an interesting structure too. These polymers tend to be helical. And you can see this reflection of that here. So that's just an example of how you can get to novel materials very rapidly through this connective approach. We've made probably 50 different polymers now using this kind of chemistry, and they all work very well. Okay, so I'm going to focus on diversity-oriented clicking today and focus on kind of lead-like discovery. Of course, we all know diversity-oriented synthesis. This was developed by Professor Schreiber. Well, diversity-oriented clicking really falls under the umbrella of DOS, but it combines the best of classical click chemistry, which had kind of been ignored really under the shadow of QAC at least, and modern suffix chemistry. So just quickly mentioning classic click chemistry, we're talking about oxidation of olefins, condensation reactions, cycloaddition reactions, epoxide openings, etc. These are the original click reactions. And so can we combine this chemistry with modern suffix technology? And how do we do that? Well, we just wanted to explore the organic connectors really and elaborate upon them. Well, we've already seen ESF, that's a double bond, an ethene next to a sulfonyl fluoride. Well, can we put an acetylene group there, or an alkyne at least? So these are unprecedented. So this became a good target kind of connector. And of course, we can substitute at this end of the triple bond too. So we have different points of diversification there. Well, the idea is if we can get to cores like this, then perhaps we can react this triple bond with, leaving the sulfonyl fluoride in touch. And indeed, this seems, feasible. So we can diversify here, and then we have a pendant sulfonyl fluoride. Well, of course, if we have that, we can go away and do suffix chemistry and build up a library, like a fragment-based approach. Or we can even do bioprospecting, because these sulfonyl fluorides are ultimately electrophilic, and when they're in the vicinity of their partner protein, they can click together and form a covalent bond. So like activity-based profiling, and this has been demonstrated by Jeff Kelly, Sharpless, and Walling groups and Lynn Jones's group, etc. So this is a really powerful technique. So how do we make them? Well, it wasn't too challenging. We decided to go from the acetylenes here, the terminal acetylene, and try to trap it with an electrophilic source of SO2F or an equivalent of that. Well, we had two methods. The first method, butyl lithium deprotonation at -78 and trap with this fluoryl sulfonic anhydride, which is not, you have to be very careful with that reagent, but you can make it, and it is fairly stable, but you can actually, you can quench it very nicely and get this sulfonyl fluoride. The second method, we just trap it with SO2 and then oxidize it up with an FSI. And these methods are complementary. This method A works better with electron deficient alkynes or just building blocks, and this works better with electron rich. So we go away, we've made a number of these now, and this is just a snapshot of what we have made. And generally we can get them on gram scale, certainly if we can scale them up at least. I think the SO2 chemistry is probably a little bit easier to do and manage. So we've really explored quite a lot, and they're pretty good yields, you know, in the 60s to 80s. That's manageable. I wouldn't say that's a click reaction yet though, however, but at least we have access to the substrates now. So what about the chemistry? Well, we want to focus on that triple bond. Well, it's classic cycloaddition chemistry in tribute to Professor Huischen. So we went away and investigate these imidol chlorides, which are precursors to nitrile oxides, and we know that nitrile oxides are incredibly reactive 1,3 dipoles. And in fact, they're so reactive they often dimerize. But in this case, at room temperature, they seem to react very nicely with our SAFs. We call them substituted acetylenic sulfonyl fluorides, so we call them SASF, SAF for shorthand. And we managed to get this kind of isoxazole product, and we got one regioisomer. You know, that's a good day. So that really is a kind of fits one of the, at least achieves one of the click criteria. And the yields are pretty good in general. You know, one or two were falling off, so I wouldn't say that was a click, but in general they were pretty good. Purification was very easy, often just a quick wash and a filtration. And as you can see, the simple mechanism or proposed mechanism is here to explain the regiochemistry. So I'd say this is en route to a click reaction, perhaps not of QAC standard, but nevertheless, we're getting some pretty diverse compounds, and we've got some nice crystal structures to corroborate the regiochemistry. Well, I'm going to fly through this because I don't want to labor on the chemistry part too much here, because these are known transformations. But we can do the same kind of chemistry with these nitrilamines. Again, 1,3 dipoles, and you can see a mechanism. You can generate this nitrilamine. It does a nice cycloaddition, and yet again, we get a single product, a single regioisomer, lovely yields, very easy purification. So we're getting these nice heterocyclic cores, and of course, they've all got these pendant sulfonyl fluorides, all unprecedented. So interesting compounds to explore. What else can we do? Well, we've already mentioned, well, sydnones have already been mentioned earlier on. Well, we couldn't forget them either. So Andrew, my colleague, went away and investigated the chemistry of sydnones, and again, we got a single regioisomer. We get, I mean, it's the same old story, relatively good yields, generally quite high, easy purification. So I do think this qualifies again as a click reaction. Even with iodo sydnones, we get some nice products. In this case, we get the 4-sulfonyl fluoride with the 5-iodo group. Now, of course, you can imagine you've got two handles here, which one could perhaps diversify from this core. So this gives another opportunity to you know, build a library. And again, the yields are pretty good. And most importantly, the regiochemistry, the single regioisomer. Of course, we couldn't forget munchnones. We've done the sydnones. So in this case, we get these pyrols with 3-sulfonyl fluoride substitution. Yet again, I'm not going to say it, but they were pretty good reactions. But my favorite, of course, can we take an azide and do classic Huisschen kind of chemistry? Well, it turns out yes. So at room temperature, you can react the SAF with this benzylic azide, and unfortunately, we get a relatively you know, the regioisomer, it was favored to this regioisomer, so it was a mixture, but it was favored to one rather than the other, but the yields weren't great. Unfortunately, when you heat the reaction up, you lose that regio selectivity, but the yield does improve. Of course, that's not perhaps a biggest problem, but it certainly doesn't qualify as a click reaction. But it turns out if you add this rhodium catalyst that was developed by Zeng, etc., and sorry, et al., it turns out you can actually get really good yields with a single regioisomer. So you get this compound here, and just to show you that we explored that library, sorry, that reaction with a number of azides, and here's just a snapshot of what we got out of it, these 2,3 triazoles with 4-sulfonyl fluoride functionalization here. I really like this reaction because in theory we can take any azide, which are readily available now given the work of Jia Dong in Shanghai, who's developed this beautiful diazotransfer reagent. I mean, we can access thousands of azides a day using this technology. So in principle, one could think about making huge libraries of sulfonyl fluorides, with triazoles, and see, explore that chemistry. So I think this is a really nice click reaction. So here's just a summary of some of the transformations that we explored from our SAFs. We've got a number of different core structures. I think that would qualify as diverse, in terms of at least, I guess, lead-like or drug-like kind of structures, diazepines here and dihydropyrules, etc., thiophenes. So I haven't talked about them all. I just wanted to give a snapshot. But this is just deals all the chemistry too. I shouldn't forget that. So there's lots of chemistry to explore. We haven't explored it all, but I think it sets the ground for SAFs as a quality hub. Now, of course, that's just the first step. We have to mention the suffix part because they've got the sulfonyl fluoride dangling off the core there, which is very useful. So in this case, we developed this array chemistry. And this chemistry could be done in 96 well plates. So we took a selection of our library. We made multiple plates with a different selection of different cores and a selection of aryl acyl ethers using classical suffix click chemistry, dBu catalysis at 20%. Room temperature, we put it on a rocker plate overnight, and we come back to the lab the next day, and we just TLC these plates because the reactions are so reliable, this spot to spot, and most of the time you see complete transformation. In the odd case where it's ambiguous, you can run away and do some LCMS, and then you can separate that way. But in generally, TLC is good enough to monitor these reactions. Of course, you can go away and screen those compounds directly from the plate, really. We kind of did that. We actually screened a whole collection of our library. In this case, I'm just going to focus on these iodoperosols, and just to show that we can achieve function through synthesis from these hubs. And in this case, we discovered a new kind of family of antibiotic, I suppose, or against Staphylococcus aureus. These members were quite fairly potent against MRSA. So we haven't followed this up, but we just wanted to demonstrate that we can get functional molecules that have benefit to society potentially. So that's really the cycloaddition chemistry, and I'm going to switch course now and talk about some unpublished work. This is our, I guess, microaddition pathways. Of course, we didn't mention that mode of action, but of course it's it's an obvious thing to look at. But again, what we find, depending on the nucleophile, we can actually get very selective reactions. And these reactions tend to be very fast, within 5 minutes, and again, almost zero purification required, just a quick filtration, if that. So just a snapshot here with secondary amines. These reactions go beautifully fast, and we tend to get the E diastereoisomer with a range of secondary amines, and these are very nice and corroborated by crystal structures here. I want pretty good yields, and again, scalable. But in this case, we can then switch to triazoles, and in this case we get a switch in the stereochemistry. In this case we get this kind of Z conformation, and again that was corroborated by X-ray. You can see this triazole is twisted out of the plane there, so I guess that helps explain the stereochemistry of the preference for the Z diastereoisomer. Of course, we can heat these up and we get the same product, so I think these are thermodynamic products. Again, at room temperature, we can go away and do this chemistry with carboxylates, and again, we get single products. And these were very good. In fact, generally, these went complete conversion. Isolation, you lose a little bit of product, but these were very, very clean. And these are, again, very unique products, very unprecedented. And you can think about having two electrophiles you could play with, perhaps as capture agents, as for looking for bioprospecting. So maybe we'll explore those at some point. But again, this is work in progress. And of course, it wouldn't be right if I didn't mention the suffix reaction. Depending on the substituent, of course, the reactivity at the sulfonyl fluoride end of the molecule does change. So you can actually tune the chemistry of the sulfonyl fluoride. In this case, with this secondary amine here, it looks like the fluorous sulfate group is more reactive than the sulfonyl fluoride. In fact, the sulfonyl fluoride is very stable. We have to really try hard to react it at this point here. But with the triazole substituent, the reaction goes pretty well in about 3 hours and a pretty good yield. So you can tune the reactivity of that end. And I think that's going to be important in the design of covalent drugs in the future. So using these cores, I think we can use them, we can discover important molecules, at least tune that reactivity. And here's a quick example of how we did that. We did a screen of our library with Dennis Wallen's group against human neutrophil elastase. Now, we chose this because in earlier work we'd shown that certain sulfonyl fluorides form nice covalent adducts to the active site serine in human neutrophil elastase, and we got a very selective compound. So we did have an idea that we may have some activity, but of course we screened a number of them, and again, the triazole groups definitely came out as the lead, the winner here. And here's a little a molecular model to perhaps explain the binding. This was a covalent reaction because, as you can see from the mass spec, we lose the sulfur. We will actually lose fluoride. So it is actually a suffix reaction. It's not displacing the triazole. So this is quite an interesting lead for us. Right, I'm going to finish off by talking about some very recent work that we published. We called it Accelerated Suffix Click Chemistry. It's just an improvement on existing Suffix, really. Suffix is a great reaction, but it can be hampered by often requiring high catalyst loadings, 30, even 40% sometimes. And generally, you need to make the aryl silyl ethers, which isn't a big problem. You know, they're easy to make. But if you want to do a huge library synthesis, and it adds an extra step. So if we can remove the burden of making an aryl silyl ether and reduce the catalyst loading, then it's going to be a good day in the lab. Well, we had some idea that arginine residues may accelerate suffix reactions in biological, biological systems, or biosuffix, if you like. So we thought we should explore some, kind of guanidine style catalysts. So we did a quick screen of available catalysts, and we chose this really notoriously slow suffix reaction between this sulfonal fluoride and this aryl silyl ether here. Now with DBU, this is very slow. After 2 hours, you only get 17% conversion. This is 20 more percent. Well, it turns out the winner here is BTMG, or Barton's base. This goes like the clappers, I'd say, really quickly. You get a complete conversion within 2 hours, and in fact, far quicker than 2 hours. So that's a winner. So we just decided just to take Barton's base and run with it rather than look for a better version. So this is where we are. We found out that we can lower the catalyst loading to 1 more percent and get the same yield within less than 5 minutes. So this is great, of course. But we also found that I should say we can remove the silica, the silyl ether. So it turns out with a catalyst and an additive such as HMDS or trimethyl silyl alcohol, this anhydride here, they all work pretty well, but HMDS really comes out on top. And this has been observed by New et al. in some early work too. So we think that it probably formed, well, HMDS is known to form silyl ethers in situ, so perhaps that's what's happening. But in the presence of 1 more percent and, well, 2 equivalents of HMDS, this reaction is incredibly fast. That's hence why we call it accelerated suffix. But it doesn't just stop with simple aryl ethers, phenolic groups. We can actually do this chemistry now with alkyl alcohols, which, this is notoriously difficult with sulfonal fluoride. It's very, very slow. So we do have to bump up the catalyst loading, but these reactions go incredibly well. So now this has really expanded the scope of suffix, at least. So hopefully that's removed the need to make the silyl ether, lower the catalyst loading. And one of the bonuses is the side products from this reaction are all volatile. So you can just evaporate everything and then you get your product out the other end. Very, very clean without need for any purification. So here's a summary of this reaction. We can take a number of different substrates. We can take aromatic alcohols or alkyl alcohols, and we get with primary and secondary, and we get a nice array of products and generally very good yields. And I got to thank these guys for really delivering this chemistry, and in particular Josh, who finished it off at Cold Spring Harbor. Well, it wouldn't be click chemistry if we couldn't get a functional molecule out the other end of it. So we decided to demonstrate the power of this accelerated suffix and investigate some analogs or mimetics, I guess, of combretistatin. Now, this is a microtubulin disrupting agent. It's very, very nice. It's quite potent. But it's a natural product. There are some drawbacks with it. Can we improve upon it? Can we replace this double bond, perhaps with a sulfonate? Now, it turns out you can, and we did this in 96 well plate format again, and we just screened a bunch of phenols, and we actually discovered something that was pretty potent. Well, a number of compounds that are pretty potent, and we screened against 2 cancer cell lines. Now, it turns out we actually reinvented something that had been discovered 20 years ago. So these sulfonate esters have been shown to be potent microtubulin disrupting agents previously, but we didn't know that at the time. We actually did our homework after the event, probably the wrong way around. But nevertheless, we did discover some interesting molecules. We screened against this HCT15 and this NCI-H460 cell line. This is a lung cell cancer with a KRAS mutation. HCT15 generally has high levels of P-glycoprotein, so this is often multi-drug resistant. But as you can see, our compound, and I'll just focus on the bottom end here, P50, it's pretty potent. It's in the same vicinity or region as Taxol or colchicine in non-multi-drug resistant cell lines. But as you can see, Taxol in the resistant cell line really falls off the edge here. So this is Taxol resistant, whereas our compounds maintain or retain their activity. So in a couple of synthetic steps, we've rediscovered a family of compounds that are very, very potent. We haven't stopped there. We've actually got some nice crystal structures now with this bound to the tubulin, I should say, and you know, it's given us some insight. And this sulfonate is really important. So it's not just a benign connector; it really is an active. It forms some nice contacts in the crystal structure. So on that note, I'll just give a quick summary. We've introduced diversity-oriented clicking as a useful tool for discovering functional molecules. We've developed SAFs as a versatile class of connector, I think. We've synthesized now, well, more than 500 of these kind of sulfonyl fluoride ligands, so we've got a decent sized library now. We've identified several lead structures already, and we have many more in the pipeline through collaboration. And we've developed accelerated suffix, which I think is a significant improvement on classical suffix reactions. I think it's just simpler, it's quicker, and hopefully it'll be an important contribution. And on that note, I will thank you for your attention and thank some of the funders for this work. And happy to take questions.
Thank you very much, John, for a beautiful lecture, an overview of what can be done with Suffix chemistry, and also what precedes the Suffix chemistry, actually, which is also quite interesting. Then it was also about kissing and sleeping beauties. So thank you very much. So I was actually amazed at those Michael additions. So you worked with nitrogen nucleophiles having the sulfonyl fluoride present. So as long as the proper catalyst is not there, it will not react with the sulfonyl fluoride.
That's right. They're really stable. I mean, you can even heat them, and the sulfonyl fluoride remains intact. Same with aromatic sulfonyl fluorides. They're generally pretty stable. You have to activate them. So that's a nice reactivity gap there between. So I guess that's what makes them valuable connectors, really. That you can unleash the reactivity with using catalysis.
Yeah, I thought of sulfonyl fluorides some time ago as some exotic reagents, but it's really amazing to see what you can do with them. I'm looking at a list whether there are some questions. Let me see. So here is one: the installation of the sulfonyl fluoride group on a terminal alkyne, deactivating the alkyne triple bond towards A sites, click reaction, but provide a suffix opportunity. Please comment. Now it's not entirely clear to me what is meant here.
No, I don't understand that. The sulfonyl fluoride would certainly deactivate the triple bond, hence why the click reaction, the Wheatstone reaction, the cycloaddition works. If that's what is meant, and of course, once you've clicked it, then there is the opportunity to do suffix subsequent to that, which we have shown that's possible. So if that's the question.
Yeah, I guess so. And another one here. Thanks for the great talk. You mentioned the sulfonyl fluoride is stable enough, and you showed a case with antibacterial activity. I wonder if this motif could serve as a bioisoster to any, say, CF substituent.
Yeah, I think so. I mean,
yeah.
Oh, is that the?
Yeah, I think that's the question. Is a sulfonyl fluoride could it act as an isoster of the CF bond in general?
It's a very good point. I don't see. I mean, it's stable enough, so I don't think. I don't see why it's worth examining it, investigating that. I should say that on a slightly different, on a tangent here, that the fluorosulfates, so you take the phenolic group and you put an SO2F group on there, they're very stable too, and they've been shown to be good in late stage kind of functionalization, and they prevent glucuronidation, for example, at that site, and they're stable generally. Under certain instances, they can undergo covalent capture. So they are electrophilic, they are potential covalent grabbers, but they're also very stable. So hence why they're called sleeping beauties. They're only, you know, that's really Barry's terminology. But really, it's a great description because they're only awoken under the right, you know, kind of right kiss, I guess. But yeah, I don't see why not. I don't see why they couldn't be used as potential bioisosteres. But you have to remember that they are potentially reactive too under the right circumstances. So.
Yeah. So I think there's a related one that is sulfur fluoride hydrolysis in vivo not a concern, but I think you just answered that. So the bond apparently does not hydrolyze.
Not easy. I mean, it's if it does, it happens very, very slowly. So no, they're biocompatible. They have to be activated to undergo, hydrolysis.
Yeah.
And they're unusually stable.
Yeah. That is clear to me now. So another one is, if the products of the Suffix reaction can be bioactive, should one be careful when thinking about using it just as a click reaction? Or in other words, I think, how careful do you have to be with the click products in general, I guess, as they may be bioactive?
Yeah, I mean, that's a good point. So like I mentioned earlier on, so there's two aspects of this. You can click your hub and at the triple bond and still have the pendant sulfonyl fluoride, and they can be used for covalent capture of proteins under the right environment, as we showed with human neutral elastase. So yes, that is a concern. So I think they have to be matched correctly. So that is definitely a concern. But one, of course, can then you can do the click chemistry on the sulfonyl fluoride. And of course, you can remove the sulfonyl fluoride group and put it like on a methyl ester or something if you want. You know, you can use that as a discovery tool at least. So you can actually remove the sulfonyl fluoride and use it in discovery, but then reverse engineer if it is a problem. So I don't think the sulfonates, the sulfonates are problematic, or the sulfonamides. I mean, they're used everywhere. So one could think about, in that context, use the reactivity to discover, but then reverse engineer it out if necessary, if that makes sense.
Yeah.
So you're not beholden to it, I should say. It's just a useful discovery tool.
Yeah. Okay, I don't see any more questions, but maybe Hannes, are you still there? Could you also show up, so to speak? Maybe there are some. So just to conclude this webinar, some general questions to ask. So I think click chemistry does exist in its current form now for like 20 years. What is going to be discovered in the next 20 years? Is this only the beginning? Was this the tip of the iceberg of what? How do you both look at this? Who wants to go ahead?
Well, I can answer. You know, generally, there are other click reactions in the pipeline from Sharpless lab and my lab and other labs. So I think maybe matching the fidelity of QAC is going to, that's going to be really challenging, right? But even if it's almost as good, or 10%, a little bit not as good, but almost, I still think there's still mileage in that. So I think there's a long way to go with click chemistry, yeah. And there's plenty of there's plenty of chemistry that can be exploited in a kind of click context too, or converted to a click reaction. So yeah.
Yeah, this is like a very modular approach that you showed, but that you also see in other reactions. That is definitely something that we will see more, I guess, in the years to come. Yeah, also for different types of reactions. And for cyclonation, go ahead, Hannes.
Yeah, and I think in click chemistry will continue to fuel bioorthogonal chemistry for sure. Even though it needs to be, I keep saying that actually there's a difference. It's click chemistry, it's bioorthogonal chemistry, and there's also in vivo chemistry. And this is sometimes, that's at least my experience, confused. Right? So just for example, I keep saying the game changer for us is the tetrasin ligation, but I can easily think of many applications I would never ever go with a tetrasin.
Yeah.
It really depends on the application. And,
So
speaking of bioorthogonal and vivo chemistry, I think that has just started.
Yeah, I agree. I mean, applications in material science, they are very different from biomedical applications. They require different things. Also the costs involved to make compounds or develop compounds, that is very different, of course. Yeah. There was one question that is not so much related to quick chemistry, I think, but it is also a general question that you may have a good answer to. Let me look it up. It said, so what is the most common misconception about organic chemistry?
That's a good question.
That's a very good question.
It depends who you ask. I mean, I'm surrounded by biologists at the moment at Cold Spring Harbor, and they have the idea that it's probably easy, or at least on the time scale of biology, it seems easy, of course, because our experiments can be done, I guess, relatively quickly. So that's a misconception. It takes a lot of skill and bravery, and really to work with some of the reagents in click chemistry. I mean, the early days of azides, of course, that was quite brave. And some of the sulfur fluorides and other agents, I mean, there has been tarnished by, I guess, phobias, because they are alien kind of species, really. Diazo groups and things like that.
Yeah.
Totally agree. It's this. If you can make one molecule, then it must be easy to make 10 more. just add a carbon here or have another nitrogen there. So you did it once. Now what's the problem to do it 10 more times?
Yeah. But indeed, that I also encountered one of the misconceptions indeed between colleagues from other fields is that we can make things easily, and that perhaps it also is due to ourselves. I mean, there were also times, I remember one of my former advisors, Casey Nicolau, saying, well, we can make anything, and, no matter how complex we can make it. But I've been working on this very complex natural product myself, and now I sometimes start lectures with it, telling that I was very proud at that time. Many reactions in a row, but if I look back at it, it was a linear sequence. There were no catalytic reactions. The atom efficiency was, atom economy was terrible. So we can still improve a lot on the way we do chemistry and synthesize molecules in terms of the diversity that we create, and on the other hand, also making reactions more sustainable or making products biodegradable. There are so many aspects, I think, that still could be improved, even apart from the synthesis. That there's still a very strong need for synthetic chemists developing new chemical reactions, I would say, based on different concepts. Now, maybe that is a good conclusion to close this symposium with. So I would like to thank all the people for attending. I see that the number is also slowly decreasing, but we are still at 100, which is definitely still a very good attendance. So thank you very much for being here. I would like to thank Hannes and John for presenting here and for writing the chapters, of course. And I also would like to thank the Thieme staff here that made this whole event possible. Thank you very much. And herewith we then close.
Thank you.
Thank you.